Microtubules are a vital part of all of our bodies, but what are they doing in our cells right now? To answer this question, let's start with the basics. Microtubule formation relies on a protein called tubulin. In our cells, there are two types of tubulin proteins, alpha and beta tubulin. These two types can combine together to form the building block of microtubules, the tubulin dimer. Through a process called polymerization, these dimers come together and form the fiber we know as microtubules. Through a similar process called depolymerization, the fibers break back into the dimers. These two processes occur simultaneously, providing microtubules dynamic properties and allowing them to adapt to the current needs of the cell.
Now that we know a little bit about the structure of microtubules, we can move on to their function in our bodies. Microtubules play a role in countless aspects of cellular function. Today, we're going to focus on three of the most important: its contribution to the cytoskeleton, its role in intracellular transport, and what it does in spindle formation. With the help of the microtubules, the cytoskeleton is able to maintain cell shape, as well as the organization of organelles within the cytosol. The cytoskeleton is made up of three different types of filaments. Of these three, microtubules are the largest component. A comparison can be made between the cytoskeleton of a cell and the bones in your body. Imagine what your body would look like without all of your bones. You would lose your normal shape and your structure, kind of looking like a blob. It would be very hard to interact and function with the people around you. The same holds true with the cell. Without their cytoskeleton, they would transform from an organized functional unit into a useless sack of water and proteins.
This brings us to the next function of microtubules, their role in intracellular transport. Intracellular transport is a mechanism by which cellular cargo, such as a vesicle or an organelle, is transported from one area of the cell to another, just like cars driving on a highway. Vroom! This transport relies on an important characteristic of microtubules, that they have polarity. Polarity means that the two ends of the microtubule are structurally different. These differences arise because one end of the microtubule is capped by the alpha tubule inside of the dimer, while the other is capped by the beta tubule inside. This polarity allows scientists and various cellular proteins to tell the difference between one end of a fiber and the other. Scientists refer to these two sides as the plus and minus ends.
Motor proteins in our cells utilize the polarity of microtubules to direct their movement. These motor proteins are responsible for carrying cargo that is to be transported around our cells. Some motor proteins only move towards the plus end, while others only move towards the minus end. Imagine a microtubule as a two-lane road between plus city and minus city. FedS only transports cargo towards plus city, and UPX only transports cargo towards minus city. One example of intracellular transport is a motor protein moving a vesicle along the microtubule from the endoplasmic reticulum to the Golgi apparatus, as shown in this animation.
The final function of microtubules we'll talk about is its job in spindle formation. You know those lines you see going to the chromatids when you look at a diagram of metaphase? Those are microtubules. The spindle is the complete package, the microtubules plus the chromatids plus these structures called centrosomes. Microtubules begin forming at the centrosome and polymerize and depolymerize in many different directions until they attach to a kinetochore and stabilize. Another proposed mechanism of spindle formation involves microtubules forming at the centromere.
Either way, the microtubules eventually organize into the bipolar structure that we are used to seeing. In this organizational scheme, the minus ends of the microtubule are bundled at the centrosome, and the plus ends of the microtubule are located at the centromere. The depolymerization of microtubules in the spindle structure then helps to separate sister chromatids into daughter cells during cell division.
Sadly, we are out of time for today. We hope you enjoyed learning about microtubules with us. Join us next week for an exciting glimpse into the world of
Sand dunes!
1, 2, 3.